Do you just go by Doc or how?
Doc.
Okay,
Yeah.
Doc Edge from QCB, and he's going to talk about using genealogies to understand complex phenotypes. So this is something that I think several of us in the division have toyed with in the past. Paul being probably the person most directly related to those. So lots of interest in this room. So thank you very much.
Awesome. Yeah, thanks so much for having me. Really nice to be here. Just by way of introducing myself a little bit, so I'm over in QCB, as Dave said, on the University Park campus, and I'm a population geneticist by training. So I do sort of broad, different sets of projects in evolutionary genetics. So this is all the stuff I'm not going to talk about today. And then that's what my lab members do. I drink a lot of tea and occasionally post a meme or two or something. When I normally come and just give a talk to an audience I've never talked to before, I often talk about forensics work. So if you have any interest in forensic genetics, I'm really interested in how forensic genetics, if you view it from a sort of evolutionary genetics perspective, there's a bunch of big privacy holes and other kinds of issues that they're not necessarily thinking about. So I do a lot of work in that space, but I'm not going to talk about it at all today. What I will talk about is what Dave alluded to. There's going to be a lot of tree work ahead, thinking about coalescent trees or genealogies in particular. I like this sign both because it's accurate, and this work is also very much in progress, as a sort of construction sign sort of connotes. So the reason population geneticists care about trees is because trees are a really nice way of structuring genetic variation. If I have a bunch of haplotypes and derived alleles denoted as here, I can, if I assume that every derived allele has only appeared once in the history of the sample, then there's some tree that connects all the haplotypes to each other. This is a tree of sort of common descent. So where these little tips run into each other, that's where they share a common ancestor. They share they're copies of the same haplotype at some point back in the past. And this genealogy sort of tells you the different partitions of the haplotypes that are possible to have, derived alleles on them and so forth. So it's this really informative way of thinking about how genetic variation at one locus can look. And if you think about, that's a non-recombining segment there. If you think about parts of the genome that can recombine, you actually need a more general structure to be able to describe it. And the structure which comes from your colleague, Paul Marjoram here, he sort of invented this in 1996. This is called the ancestry recombination graph. And it's a lot like that tree I just showed you, except instead of being sort of a pure death process going back in time, it's a birth and death process where the birth are recombination events that sort of mean that one side of the haplotype, for example, follows one path up the tree and the other follows the other path. So this arc structure is enormously richly informative. All of the mutation and recombination events that could possibly affect genetic variation in a present day sample are encoded by the true ARG, which is of course unknown. We can also represent the ARG as a sequence of trees. If I look at any one place in the genome, like one base pair, for example, it's not going to be a graph, it's going to be a tree. I guess it is still a graph because trees are graphs, but it's going to be describable by a binary tree. So the way we represent the ARG in this way is every spot mutation has a tree, and as you move from place to place, there might be recombination events, and those can change the topology of the tree in a well-defined way. Yeah, so essentially we use this tree. It's called a tree sequence sometimes when you represent it this way. It's not exactly equivalent to the full ARG, but I'm going to completely elide that distinction today. I like these a lot. My lab logo has a tree sequence in it. But yeah, I'm going to use ARG and tree sequence totally interchangeably to basically mean the set of trees that describe describe the genealogical history of the haplotypes in your sample at any one spot. So I already said this, the key thing about ARGs is that they encode every mutation or recombination event that can affect variation in present-day sample. So if you care about genetic variation, there's sort of your sufficient statistic for any possible analysis you would want to do. And that makes us, as population geneticists, sometimes feel a little bit religious about them. Here's Adam Steeple. I believe in one ARG, creator of heaven and earth, of all things, visible and invisible. Of course, a little bit tongue-in-cheek, but I think it's catching something real. So a really exciting thing that's been happening over the past 5 or 10 years, we can never know the true ARG for a sample unless there's very specific experimental circumstances that let us infer it. But if you have just a sample of unrelated humans, for example, how do you try and get it? It's extremely difficult to estimate the ARG. It's a massive space of possibilities to relate a set of haplotypes. But there's been a lot of progress in the last 10 years. ARGweaver came on the scene about 10 years ago and really pushed things forward in terms of being able to infer not the true ARG, but an approximation to it with this really satisfying. You can get like pretty good posterior samples from an approximation of the ARG called the sequential Markov coalescent, which Paul has also written a lot about. Rent Plus came out about 5 years ago to essentially no fanfare at all. But it essentially, if you know phylogenetic tree inference methods, it's doing UPGMA in Windows, and then a bunch of computer science tricks to sort of make things work, resolve the discrepancies that come from that process. And then in the last 2 or 3 years, a lot of excitement has come from these two methods, Relate and TS-Infer, which came out in the same issue of Nature Genetics in 2019. And these are the first ones where you can really get an approximation to the ARG with thousands, or even in the case of TS-Infer, potentially tens of thousands of haplotypes being connected. So the big question is, what can we do with this new way of representing, or it's not a new way of representing genetic variation, but it's a unprecedented level of access to an approximation of the underlying ancestor recombination graph. So that's what's going to motivate my comments today. So today I'm going to tell you about 2 projects in my lab that try to take advantage of these new methods. First, this is work by primarily my student Dan Peng and also Obadiah Mulder, where they're trying to combine GWAS information with ancestral recombination graphs to learn about the history of complex traits, learn about their evolutionary history. And then the second part, which is led by my postdoc Vivian Link and in collaboration with both Nick and Charleston here, is actually trying to use the estimated ARGs to identify the trait-associated loci, sort of to do the GWAS itself. So first project, what can we learn about polygenic trait history? So I mentioned I'm coming from evolutionary biology mainly, and so we really care, right, about how traits change within a population over time. That's sort of one of our main things we care about. So this is what I'm showing you here is over time archaeological reconstructions of height from skeletal remains within Europe. So archaeology is one tool for this, but we don't always have, for every trait we might want, we don't have the ability to reconstruct things from archaeology, or we might want to just figure out the best way we can use contemporary genetic data to come at this same kind of question. So for example, a more biomedical question that would be harder to get at with skeletal remains, say, is the thrifty genotype hypothesis, which maybe some of you have heard of. It's this idea that maybe we understand something about the risk of metabolic conditions like type 2 diabetes by thinking about how the sort of food environment we're in now is a mismatch to the one we've adapted to in some sense. So the idea is, food becomes a lot more easily accessible with agricultural and then especially with industrialized food. And maybe something about that transition is useful for understanding differences, say, in diabetes risks around the world. So that's just, if you can understand something about how a particular trait evolves, there might be biomedical applications as well. So I think everybody in the room knows what a GWAS is, but GWAS is really going to be the key for us. So of course, for anyone who doesn't know, in a GWAS, we just take genotypes, SNP genotypes around the genome, associate with them with some phenotype, we estimate a slope, we get a bunch of p-values, hopefully find some loci that are associated with complex traits. And so of course, there's been a huge explosion in people's interest in GWAS over the years, and there's been a massive effort devoted to it. And one thing I want to do is just slide in here and try and steal some of this information to answer some evolutionary questions maybe. So Charleston just has to have this step out, but he actually co-led the first really important paper that used this strategy of trying to take GWAS hits and learn something about how a trait might have evolved. And a lot of this work has been on height, but there have been a bunch of methods for this over the years, mostly focusing either on differences in population allele frequencies or sort of simple summaries of genetic variation around the loci that are tagged in a GWAS. So our question, I think, is can we use the ancestral recombination graph to get a better handle on how at least the polygenic score has changed over time? We first don't have the trait itself. We have the contemporary polygenic score. I'll say more about that in a second. So the tool we're going to use is the marginal trees that describe genetic relatedness at a particular location in the genome. So this is like one tree from the tree sequence or from the ancestral recombination graph. We call these trees coalescent trees. Coalescent trees are trees that come from a particular data generating process that might describe a gene genealogy. So the way this representation, right, is we're in the present at the bottom and we go up into the past. And we're sort of tracing the history of the sample back in time, recording when individual haplotypes share common ancestors. And the key thing here for my purpose today is that the rate at which these coalescent events occur, these mergings of lineages, is proportional to this terribly notated quantity, if you're not used to the notation, little n choose 2 over big N of t. So little n is the number of lineages that have not yet coalesced. So it's like the sample size, essentially, at least initially, until coalescences start happening. And big N of t is the population size at time t. So the reason that notation is bad is little n is not like an instance of big N, which is random. It's just something else. Yeah, so the key thing is we've got the population size here that's determining the coalescent rate. So if I can measure the coalescent rate back in time, I'm getting information about how the population size has changed over time. And people, in fact, use this intuition all the time to try to do just that, to estimate the population size. But what I want to propose is that if these gene genealogies or these coalescent trees encode population size history, that means they also encode, albeit quite noisily, allele frequency history as well. Because allele frequency history is just the size of 2 different subpopulations going back in time, right? The carriers of one allele and the carriers of some other allele. And so that means we can use this intuition to estimate the history of the population mean level of a polygenic score. So polygenic scores, I think, are quite familiar to people in the room. I'm going to focus on this special case where it's just a linear combination of with an individual, we're thinking of a linear combination of their genotype. I'm thinking about a population mean level, so it's a linear combination of the allele frequencies going back in time, where the weights are the effect sizes, say from the GWAS. And you might think of the changes in this quantity over time as being related in some way to the changes in the trait value in the population. Kind of caveats to that, right? You have to assume no epistasis, no gene by environment interaction, no dominance, etc. And then especially that the way in which we're tagging the causal variance is sort of consistent through time. And we know that that's not really true, right? We know that the reason we know that is we know if we look at contemporary populations and we apply a polygenic score from one to another, it doesn't quite work, it sort of kind of works. And the same thing is going to happen as we go back through time. That said, so there's a ton of caveats here. That said, I won't talk about it much today, but there are patterns of change that you might observe in a polygenic score over time that are, say, inconsistent with neutral evolution. So you can still learn something about selection, even if you're not directly reading out, this is how the trait has changed over time.
So just a little bit, some questions about that last there. Is that you're speaking out about these alleles, but do you really mean haplotypes? And since you're talking about recombination graphs locally, and you're saying that the haplotype tags a causal variant all the way back. So, or are you saying that you actually, I mean, the other way to say that is that you just have the causal variant and that's consistent across time, right?
Yeah. Yeah, so that's a great question. And I think, so all the simulations I'm going to show you today are as if we have the causal variant itself. And so I'm just looking at the marginal tree for the causal variant. I think it would be really interesting to do something where you sort of average over uncertainty and where the causal variant might be in the haplotype. And so use information from a bunch of different neighboring trees.
Okay.
I haven't done that yet.
Yeah.
So I've written about the caveats on this in other places. So just to concretize this, I'm going to show you this as a sort of example. It's like one representative simulation where I have a population polygenic score, and in this simulation, everything is causal variance. This is actually how the trait is changing. All my assumptions I had to make are satisfied. So the trait's sort of evolving neutrally in this case, and then there's selection for some period in the past, and it goes up quite a bit, and then it evolves neutrally a bit after that. Neutrally, it just means no selection in the trait. So I'm going to take two different approaches to estimating this. The first is really simple. I'm just going to look at the tree and look at the allele frequency in the actual tree. So I'm just looking at the ancestors of the sample. And then the second one is, I'll explain both of these more fully. I'm actually going to try and compare the coalescent rate across the two backgrounds. So the first one, allele frequency in the ancestors of the sample. So like if I really know this tree, right, then these branches as I go back in time are ancestral lineages to the sample. So if I have this sample of 6 in the present, and I care about the allele frequency at some time t, represented by this dashed line, then my best estimate of the ancestors of the sample is just 2 over 5 here, because I've got 2 orange lineages and 3 green ones. So 2/5 of the ancestors at that time carry the orange derived allele, and that's it. So it's super simple, right? So this works. If you think that this allele is evolving neutrally, this is like exactly the right thing to do, because the ancestors of the sample are just a random sample from the ancestral population at that time in the past. But if there's been selection on this locus, that's no longer true, because the individuals who are ancestral to the sample are not like the ancestors back in the past. That's sort of another way of saying that's just what natural selection is, right? Is it's like biased sampling from the ancestral population. So when we look at the result in simulation, again, here's my my true sort of path I'm trying to estimate. We see exactly this intuition borne out. It looks really good. This is like in human terms, this is like, say, 10,000 years or something, this period. So it's like a decent amount of time, we're looking great. And then once selection occurs, basically it underestimates the strength of selection. Because, you know, if you imagine this is height on the y-axis and there's been selection up on height, what that means is that the ancestors of the sample are actually taller than the population that they're from. And so you underestimate the strength of the change in the ancestral population itself. But then it sort of kind of generally captures the contour after that. So how can we fix this? Well, one way to do it, we have a few ideas, but I'll just tell you about one. One way to do it is you actually break the tree into two subtrees, and you use that intuition I gave you earlier. So you basically say, okay, I've got a population of derived allele carriers and a population of ancestral allele carriers. It's a little funny because I'm talking about the haplotypes, right? In some cases, those haplotypes are mixed up in the same individual. But we're going to be like competitively estimating those sizes by looking at the coalescent rates on the two subtrees. So one way to do this, there's multiple ways to do this, but one way is I pick 2 time points on the tree, and I notice that there's been a change, say from 3 lineages down to 2 over that time point. And then I can use this approximation, which comes from a paper from about 10 years ago, that says, okay, given this amount of time has passed, and given the number of lineages I started with and 0, here's the expectation for how many lineages I expect to see. This has terms for both the time passed and the sort of harmonic mean population size over that period. I invert this expression, use it to estimate these 2 quantities and put it together. It's going to be quite noisy because we're essentially relying on small numbers of exponentially distributed random variables to estimate each of these quantities. By the way, this is an approximation. The exact value is numerically unstable for large trees, which is why we use the approximation. So here's the old one. And if I do this new method that I'm suggesting, it's really noisy, as I told you to expect, but it does actually nail this period. The sort of strength of selection is right. This is with the true trees, though. And by the way, we're thinking a lot about how can we sort of use the merits of both of these, trade them against each other in some way. Because, you know, the proportion of lineages is nice, but it's biased. We've got a real bias variance trade-off, like literally here. So we're thinking about how to deal with that. This is just one run, but if I did if I showed you an MSE from 100 runs, it would be really concordant with this. That's all true trees though, right? That's all well and good, but what if I have to estimate the trees? So we're thinking a lot about this too. And we have all these different possibilities. As we go down this list, we can feed in bigger samples, but they're also all using different methods for estimating the branch lengths. I'd say in particular, this one really just uses like Hamming distance between haplotypes. That's what tells you how distant they are. These ones are essentially taking samples from a posterior that's informed by like a neutral coalescent prior. So there's a little bit of prior information being used to generate the branch length estimates. So we're going to do some simulations, and what we're going to do is we're going to stack the deck a bit. We're going to do the same sort of situation I showed you before, but with 100. We're going to do it 100 times. This is Dan's work, and we're going to try to estimate these quantities using different ARG estimators, and we're going to use approximately the number of lineages that they can take. So 20 lineages for ARG-Weaver, 200 for Ferment Plus, 2,000 for Relate, and I'll also use 2,000 for TS Infer, even though it could go up larger. And this is what you see. This is like hot off the presses in the figure from yesterday. So I'm showing you the mean squared error going back in time to the left. So this is the presence, and then there's a period of selection here. And this is the estimator that we know is not very good under selection. So even with the true trees, there's a big bump in the mean squared error that sort of relaxes when you go back to neutrality. And all the colored lines are the different estimators. And you actually see a really interesting thing, I think, which is that Rent Plus, which can only take 200 samples and was not released with very much fanfare, it's actually doing the best overall. That's a little tough to see over here on the right in the neutral period when this estimator makes the most sense, but it's actually still doing the best there. It's pretty competitive even with the true trees in that case. And if I look at the other estimator, the lineages remaining estimator, it's a little bit more complicated, but I still basically see the same thing, that relate, even though it's taking in the TS infer, even though they're taking in 10 times more data, relate sort of competitive with it for at least part of the trajectory, and TS infer isn't close to as good. So why is this? Why don't the bigger samples that we get from relate and TS infer help more? And I should say, relate and TS infer are amazing. And, you know, depending on what, this is like a very specific purpose, right? I guess my overall point is that it depends what you want to do with these trees, whether you want to scale it all the way up or use something that maybe can't scale, but has a different way of estimating the thing you really care about. So the intuition for why don't we do better with 10 times as many data is that, recall the coalescence rate is proportional to a little n choose 2, where a little n is essentially your sample size. So if you feed in a really big sample, all that stuff just coalesces right away. It's gone, and it can't help you with your estimate anymore. So we should really even only expect the larger sample to help us in the very, very recent past. And actually, you know, having 200 lineages, once we get back out of the very recent past, is just about as good as having 2,000. That should make us sad as evolutionary biologists in general, because we can never get consistent estimators for some of these things we care about. But it sort of explains this pattern. We've used this a bit to look at phenotypes, and you know, if we look at height, for those of you who've been following this stuff, there's this story about selection on height, is it there or not, and it depends, it's a stratification, a quadratic score, yada yada. And we recapitulate what everybody else sees, which is that if you look at the giant meta-analysis, it does look like there's selection, but it's probably stratification. Because if you look in UK Biobank, it's not there. Charleston has a nice paper from, I think, last year, where he took this method, among others, and found, compared Sardinians and other Europeans, and actually found some evidence when you look there that there's sort of divergent selection maybe on height. But I'd say this particular story is still a bit up in the air. That thrifty gene hypothesis I mentioned, it looks, if you do the type 2 diabetes polygenic score and feed it through this method, it looks kind of consistent with what you'd expect from thrifty gene. Whereas if you think type 2 diabetes risk might have something to do with, you know, say, being able to get a lot of nutrients out of whatever you eat, we see a decline in this right around the dawn of agriculture, which, like, kind of neat. I don't know whether this is super preliminary. I don't really know. I'm not telling you to believe this, but I'm just telling you that there's maybe some interesting evolutionary stories we can look at with this method.
How about when you apply your models to traits that you know don't have selection, to go back to the initial assumption that you had, sort of the estimation in traits that don't have selection versus estimation in traits that do, like a late onset cancer or something like that, and then you go back, right? Because there's an added, okay, is there selection? Is it consistent selection? All of these issues here versus a trait that you know is probably late onset and won't have that issue.
Yeah, that's really interesting. So I should say, in terms of actual traits I've applied this to, really small number. Okay. But yeah, we're thinking about, we have this problem. An interesting thing is that I didn't talk at all about our test for selection, but it works way better with the estimator that's bad for selection. So you have to kind of do this thing where you use that one to test for selection. If you don't find it,
Oh, I see.
that one's
Okay.
probably safe. And if you do, you probably have to switch to the other one. So we're sort of still figuring out that pipeline. And I'm kind of trying to get clear on that before I apply it to a bunch of traits, but I very much want to do that. So in the last 20 minutes or so, I'll talk about this other project that Vivian Link is leading, where we're trying to find trait-associated loci using ARGs. And this is in collaboration with some folks you know, with Charleston and Chao Chi Fan and with Nick. It's been a lot of fun to work on. So if you ask, can we use ARGs to identify trait-associated loci? In some sense, we already are, right? Because if you do a GWAS, for example, what you can think of it as you're testing a branch in the ARG. Anytime you test a mutation or SNP for association with a phenotype, this is, there's a cartoon where this phenotype is sort of enriched under this branch where the mutation appeared, and that is what a GWAS signal is, if you think of it from this perspective. But the ARG is a lot richer than that, right? We don't just have to test one branch at a time. We can do things that are sort of taking into account the full pattern of relatedness if we want to. Again, going back to, this is our OR statistic for everything. So let's take it out for more of a spin. So there were a bunch of things we could have done in terms of ways to try and use marginal trees or ancestral recombination graphs to look for trait-associated loci. And we thought, well, what is GWAS? What are some of the maybe issues with it? And we focused on a couple. One issue, less of an issue as you have larger and larger sample sizes. I should say all of these, I'm really thinking about understudied populations or maybe non-human organisms. If you throw 600,000 or a million humans at something, you can do whatever and it'll work. So in general, in GWAS, you're doing a lot of tests, right? And if you have an ARG, some of those tests are somewhat redundant because they're, from an ARG perspective, they're on sort of really correlated branches. And if you know the whole ARG, maybe you can get rid of some of that redundancy. GWAS is also not great for dealing with allelic heterogeneity. I'll explain more what that is on the next slide if you don't know that jargon. But the ARG gives a sort of complete representation of co-inheritance in a region. And so, reasons to think it might be better at that particular job. And the other thing, this is sort of a, this is not something I show you numbers on, but just philosophically, like the process of actually doing imputation and then testing the imputed variance for association, that's conceptually a little bit roundabout, right? Like we don't actually need really good, imputing the individual level variables is probably a little bit harder than figuring out the relationships that underlie those imputations. And so maybe there's a way you can kind of get around needing a good imputation panel by using the ARG instead. Because you don't, to infer these ARGs, you actually don't need like a sequenced panel or anything. So that's one thing to keep in mind. So allelic heterogeneity, this is what I'm really going to focus on. So I'll say a little bit more about it. This is just when you have multiple causal variants at a particular locus. And that's not rare, right? Back in the linkage era, we found that all the time. Linkage analysis is totally robust to this in a way that GWAS is not. And if you try to estimate it now, it looks like maybe 20% of loci for complex traits have a little heterogeneity going on. So this is not like a small issue. And what can happen, doesn't always happen, but what can happen is you can sort of confuse a GWAS because your tag SNP is in plus LD with one of the causal variants and negative LD with the other one, and the signals kind of cancel each other out. And so you can have situations like this. This is just one simulation, but Vivian has simulated a sort of causal region in the middle here, centered on this blue point, and done a GWAS around it. It's a little heterogeneity in the region. She just doesn't see anything with GWAS there. So this idea of using ARGs to maybe get around this problem is not a new idea. It was proposed in the mid-aughts several times. It was a string of papers, but it kind of went away, I think, for two reasons. One is that estimating the actual trees or the graphs really didn't scale at the time. We were talking about samples of a few dozen haplotypes, and the other is that meta-analysis really came to dominate mapping efforts. Right, we need to do everything with summary statistics, etc., because you didn't have individual level genetic data. So I think both of those suggest that now is a good time to come at this question again. One reason is that more and more often we're working with like biobank data, and we actually have individual level genetic data. Summary statistics will remain super important, right? But maybe relatively less important than they were, say, in 2010, arguably. And the other is that we have these methods that can actually estimate these graphs for pretty large samples. Relate's not getting into the full-on like human GWAS neighborhood yet, but TS infer really is getting at least close to it. So our general strategy is, if we have a sample and we know some region in the genome, we know how they're related to each other, we know how long ago in the past different pairs of haplotypes coalesce, we can build a matrix of those times. We're not going to literally use exactly that matrix, but a sort of rescaling of it that Chauchi and Nick and Charleston invented. And we're going to look at how this coalescence time pattern is related to differences between people on the phenotype. So if I find that in a region coalescing really recently is correlated with being really similar on the phenotype, that's my signal that I'm looking for. So the way Chauchi and Nick and Charleston have developed to do something like this is as follows. So if you start with I'm just taking a figure from their paper from earlier this year. If you start with a genotype matrix, one thing we do all the time, right, is we build a genetic relatedness matrix. So essentially what we're doing when we do that is taking the vector of genotypes, centering it, normalizing it, turning it into something like a variance covariance matrix, and then averaging that over all the different genetic variants that we see. So it's an unweighted average over the genetic variance. And what this paper from your colleagues does so nicely is sort of understand that there's actually trees underlying all those genetic variants you might see. And another strategy you could take is, instead of taking an unweighted average of the genetic variants themselves, you could take a weighted average of the branches, where the branches, the branch lengths are the weights. And what you can interpret this as is the expectation of the GRM conditional on the tree itself. So the topology of the branch lengths, it's the expectation over the randomness in the mutation process. So assuming mutations get placed randomly on the ARG, you can compute an EGRM. And in this paper, they're focused on the EGRM for the whole genome, but we're going to talk about EGRMs for a locus. And of course, we use these all the time to do control for population stratification or to estimate heritability. So the model is really simple. We're just going to say that the trait is normally distributed, conditional on some fixed covariance, with a covariance matrix that's partly white noise and partly described by this local level expected genetic relatedness matrix, given the local trees in the window we're looking at. So A, here is the local EGRM. And what we care about is this parameter sigma squared A, and we're going to estimate it with Remel, just like we would do a sort of heritability estimate with a genetic relatedness matrix. So incidentally, this is also an older idea. Doing it with the, not with the EGRM, but with the GRM, goes back at least 20 years. And when we do this for that same locus I showed you before where it didn't work, we find a nice peak here. It's not centered right on our causal variance. These peaks tend to be pretty broad that we build in this way, but we do find the peak. So I'm going to show you some simulations. This is just one out of a whole bunch that Vivian has done. So we're going to do 2,000 chromosomes. Again, we're going to use relate, so the samples aren't going to be huge. They're going to be human GWAS level. And we're going to have a 5 kb window. That's our causal region. You could argue how long should that be? And what I'm going to show you is 40% of the variance in that region are causal. You could argue what should that percentage be? And the effect sizes are going to come from this distribution that effectively makes larger effect sizes, or effect sizes negatively correlated with allele frequency, which is what we see in real data. So we're going to type 20% of the variance. We're going to try to do GWAS. We're going to do local GRM, and we're going to do local EGRM. And I'm not showing you this. You do have to do fewer tests. Turns out I'm not going to make a big deal of that, but you get away with a type 1 error control that's not quite as stringent as you have to use in GWAS. So I'm showing you first just GWAS and the local GRM. So the heritability, this is like the effect size essentially in the region, and it's going up quite high, right? Because our sample sizes aren't huge, but these aren't bonkers for say some molecular phenotypes. So GWAS is in gold. This is power on the y-axis. GWAS is in gold. Local GRM is in so this pink color. And despite this sort of local GRM idea being a really good idea for capturing a little heterogeneity, it doesn't really help. But when we put in the EGRM, we get really quite a substantial benefit, actually, from doing this. The EGRM, so it's the expectation of the GRM given the local trees in the window. So that's the new thing we're doing. So, why do we get this power bump? And we've tried a bunch of different window sizes and causal effect proportions. You don't always see this big of a bump. Sometimes you see a bigger one. I tried to pick one that was like one of the better ones, but not the best one.
So
yeah.
sorry, is that a test of the sigma A? Or is it? It's not a test of any single SNP or any,
yeah, it's a sigma A in the 5 kb window.
Got it. Okay,
Yeah.
so it's a single.
So GWAS is taking every variance.
SNP, and then
We count it as a hit. As you get anything in the
yeah.
anything even close to nearby, that's a win for GWAS. Local GRM is a
Testing
GRM
the
from the
region
5
given-
kb window, and local EGRM is just given the trees in that same window.
Okay.
So why do we get this power bump? And this is how I'm thinking of it right now. If this is your window for analysis, these green lines here, so it's just this one tree in the middle. If you're the local GRM, that means all the information you can use is the variants that fall inside that window. But if you're the local EGRM, you actually get to borrow information from neighboring windows because for example, like in this particular split, like this tree down here, some of these branches aren't informative about the ones over here, but like this one is. I can use any mutations that occur over here between these two haplotypes, and they inform me about the branch length over here. So I actually get a better estimate of the local relatedness from the EGRM than I do from the GRM itself, because I borrow exactly the right information from neighboring regions. So I can for a smaller window, I get a pretty precise measurement of the relatedness. So I showed everything for a Lilith heterogeneity. If we just put in one causal variant, we're not doing a lot worse necessarily. So it's not that this is specializing on heterogeneity. It still works okay, even if the architecture is sparse in the region. That's not, it depends on the Lilith frequency, and we could spend a whole hour talking about that. But across the board, it's not as if you give up all your power for sparse architectures in order to get better power when it's more heterogeneous. In terms of extending this, we haven't done anything with real data yet, but I'm really excited about the different ways we extend this. There's a whole universe of things you can do once you have this representation. Like, for example, I already assumed in my data generating model that bigger effect alleles are going to be down here on the shorter tips, but my analysis doesn't actually use any of that. So there's ways to make this even better by feeding in things we already know about, say, stabilizing selection on a particular trait. So zooming out a bit, just want to reiterate that really any analysis of genetic variation can be framed in terms of the R, and there might be benefits to that for a bunch of different types of analyses we do in statistical genetics. So I've already said that sort of GWAS and local GRMs can be framed in this R framework, and at least in the local GRM case, there's potentially substantial benefit. But IBD mapping is another one. In IBD mapping, we essentially treat haplotypes that are IBD with each other as being distance 0 from each other, and everything else is some positive distance. But if you know the whole R, there's actually a lot you can do with that. You can actually view our method as like a generalization of IBD mapping that sort of slides up how high up the tree you're willing to treat as being IBD. And with linkage analysis, it's, similarly, we're just checking, do you coalesce right now because you have you got the same allele from mom, or do you coalesce somewhere back in the population? And we don't actually track when do you coalesce back in the population. So maybe there's a way you could actually improve the power by keeping track of that, even in a family design. I have no idea. Spitballing here, right? So in general, I'm really excited about this way of thinking about things. I think it's a fun language for communication between evolutionary genetics and statistical genetics that I hope you're convinced you can go in both directions. So that's all I got. Thanks to my lab who did all this work, to my collaborators, Chachi and Charleston and Nick, and NIH funded this. And thanks to all of you for watching.
Do we have any questions from the room or online?
I have a question, if I may.
Hi, Paul. Yeah.
Yeah. So going back to the first part of the talk, you talked about the fact that when you based your results on estimates from TS-infer or relate,